The overall goal of this procedure is to systematically collect and analyze a constant series of cerebral regions symmetrically dissected from both hemispheres of the human brain, in order to assess severity, clinico-pathologic staging, and hemispheric distribution of various brain diseases.
This method can help answer key questions in the neuropathology field, providing a systematically organized collection of qualitative and quantitative neuropathologic data for detailed clinical pathologic and imaging pathologic correlations. The main advantage of this technique consists in yielding relevant data related to the pathologic processes per se, as well as the histofunctional specialized aspect of each cerebral hemisphere.
After fixing postmortem human brain tissue according to the text protocol, place the brain on a flat surface with the frontal poles directed opposite the investigator. Orient the brain to allow full and clear visualization of all cortical gyri and sulci of the entire cerebrum. Examine the tissue for meningeal anomalies, macroscopic hemispheric asymmetries, macroscopic tisutal lesions, congenital malformations, vessel abnormalities, or any other abnormalities or unusual presentations of the cerebral surface. With the frontal poles oriented away from the investigator and the superficial aspects of the hemispheres facing the investigator, take as many digital pictures as necessary to document possible macro anomalies and to account for possible clinic neuroanatomical and post-cutting considerations.